Many foods contain carbohydrates. Figure 1 shows information about 4 different foods. So here's figure 1 just below, and we have pie charts with beans, chicken, orange and rice. And on the right hand side we have a key telling us what each of the colours or patterns mean. Which food contains the highest percentage of carbohydrate? Tick one box. So if we have a look at our key, the top one is the carbohydrate, so that's the area without any pattern. And we can see that the beans and the rice both have quite large areas without the pattern. So the rice is about 75%, it's about 3/4 of that entire circle, whereas the beans is just over 50%. So we can say that rice is the one with the most carbohydrate. Estimate the percentage of water found in beans. So we've got a copy of figure 1, and beans is the top left hand pie chart right there. And we can see water is the grey shaded box, so the bottom one in the key. So if we look for the grey shaded area in the beans, we might decide to work this out by calling that angle in the centre x. So x is approximately 90 degrees, so it's about 1/4 of that entire pie chart circle that we're looking at. And as a percentage, a quarter equals 25%. Look at figure 1. Why would eating only beans provide a more balanced diet than eating only chicken? So if we have a look up at figure 1, we can see on the chicken pie chart we only have 1, 2 and 3 different patterns on the pie chart, so that's 3 out of the 4 possible patterns. Whereas on beans we've got 1, 2, 3 and 4 of the possible patterns, so we've got all 4 food groups in the beans, and that's the answer to our question. Beans contain all food groups, which is a more balanced diet than with the chicken, which only has 3 out of those 4. Sugars are produced when enzymes break down starch. What is the name of the enzyme which breaks down starch to produce sugars? Tick one box. So if we have a look at the options the question gives us, we've got amylase, bile, lipase and protease, and amylase is actually the correct answer. That's the enzyme that breaks down sugars. If we have a look at the others, bile is a liquid substance that is secreted by the liver, and it helps to break down fats into your fatty acids. Lipase is an enzyme like amylase, and it breaks down lipids, so it does a similar job to the bile, breaking them into fatty acids. And protease, also an enzyme, breaks down your proteins into amino acids. Which chemical could be used to test for glucose? Tick one box. So looking at the options, we've got Benedict's reagent, burette reagent, iodine solution and sulfuric acid. So the answer to this question is Benedict's reagent. The burette reagent tests for proteins, iodine will test for starch, and sulfuric acid tests for fat content, so this is quite often done in milk. What colour change would be seen in a positive test for glucose? So remember, this is the Benedict's reagent, which is blue to start off with. And the answer to this has quite a few options. So it can turn from blue to red, however it can also turn from blue to green, yellow, orange or brown. The mark scheme will allow you to answer with any of those colours. People with diabetes have difficulty controlling the concentration of glucose in their blood. The blood of 4 people was tested. Table 1 shows the results. So here we've got table 1, the left hand column has different people, they've been labelled A through to D, and then we have the concentration of glucose in the blood in arbitrary units. Table 2 shows the information used to help decide if a person has diabetes. So here we have table 2, and the left hand column is the concentration of glucose in the blood in arbitrary units. And on the right hand side is the conclusion. So less than 5.6 is no diabetes, 5.6 to 7.0 is mild diabetes, and greater than 7.0 is severe diabetes. Which person has severe diabetes? Tick 1 box. So we can have a look at table 2 and we see that greater than 7.0 is equivalent to severe diabetes. So we've got to look at table 1 and see who's got a value of concentration of glucose that is greater than 7.0. And that can be seen with person C, who has a concentration of glucose of 7.1. 7.1 is greater than 7.0, therefore person C can be identified as somebody with severe diabetes. So that's the box you tick. Figure 2 shows part of the human digestive system. So here's figure 2, it's a diagram with human organs on it, and we've got label Y and label X. Glucose is absorbed into the bloodstream in part X. Name part X. So let's have a look at this diagram as a whole. The large structure at the top is the liver, the next one on the right is the stomach, the smaller one in between is the gallbladder, and then the slightly strangely shaped one just below the stomach is the pancreas. Then you've got the two kind of tube structures, and the one on the inside, label X, is the small intestine. So the next question gives us some sentences, and we have to complete the sentences choosing some answers from the box. And we've got our diagram in the bottom right hand corner to help us out. The first part says some glucose is absorbed into the bloodstream against the concentration gradient by the process of blank, and we have to fill in what the blank is using one of the words from the box. So if we have a look at these words at the top, active transport is the first one, and it's like diffusion in that it's describing the movement of molecules, only this one requires energy, and this is because it's going against the concentration gradient. The next one is osmosis, and this is describing diffusion of water molecules in the simplest terms. It's the movement of water molecules across a partially permeable membrane, and it's a passive process. Then you've got digestion, so that's when you're breaking up your longer molecules, your proteins, lipids and carbohydrates into the smaller molecules that you can absorb into the bloodstream. Respiration is one of your energy producing reactions, so it's the intake of oxygen and then you exhale your carbon dioxide. And last is excretion, so this is the way the body gets rid of all of its waste materials. So again, the question is saying some glucose is absorbed into the bloodstream against the concentration gradient by the process of active transport should be your answer there, because we're going against the concentration gradient, that's the main clue. The next part says water moves out of part Y and into the bloodstream by the process of blank. So if we have a look at part Y on our diagram, part X is the small intestine, and then the larger tube that part Y is pointing out is the large intestine. Now the sentence is talking about water, that's our key clue here, and if we look at our key words, osmosis is the one that speaks about water, so that's going to be the answer down there. An animal called an axolotl lives in water. Figure 3 shows an axolotl. So here's figure 3 just below, and we can see that there's a label on there leading to the gills. Oxygen enters the axolotl's bloodstream through the gills by diffusion. What is diffusion? Tick 1 box. The first option says it's the movement of particles from a high concentration to a low concentration. And another way in which this is said is along the concentration gradient, which is something that is applicable in diffusion. The next option is suggesting that diffusion is the movement of particles from a low concentration to a high concentration. And another way in which this one is said is to be against the concentration gradient. The last one says the movement of water, and notice that this is different from the previous ones who both said the movement of particles. So this one says the movement of water from a concentrated solution to a more dilute solution, which is similar to the first one that went from a high concentration to a low concentration. So the correct answer is the first sentence, the movement of particles from a high concentration to a low concentration. And so this one's a long concentration gradient and it's the movement of particles. The second one down is the movement of particles from a low concentration to a high concentration, so this is active transport and it requires energy, as opposed to diffusion being passive. And then we have the movement of water from a concentrated solution to a more dilute solution. So this is osmosis, and it's like diffusion, just occurring with water. Describe how one feature of the axolotl's gills increases the rate of diffusion of oxygen. Use information from figure 3. So we've got figure 3 in the bottom right corner here, and we can see the structure of the gills in the purple circle. And what we notice about them is that they have these projections outside of them, and what this does is increase their surface area, which means that there's more space for the diffusion to actually happen across. So that's what you can say for your answer, that the axolotl's gills have many projections, and that this gives them a large surface area. If a gill of an axolotl is removed, stem cells in the damaged area will divide and a new gill will grow. Complete the sentence and choose an answer from the box. So we've got a box and it says adaptation, differentiation, evolution and variation. And we have to pick the right one to finish off the sentence below. So it says, when stem cells specialise to produce gill cells, this process is known as blank. So let's see what the terms that we've been given actually mean. The first one is adaptation. An adaptation is a characteristic of an organism that gives it a survival advantage, and so adaptation as a process is developing these characteristics, and it often happens as a result of random advantageous mutations. The next term is differentiation. So this is the process in a cell's development where it starts to express specific genes that give it characteristics making it into a specific cell type, so like a muscle cell or a nerve cell. Then there is evolution. So this is something that happens over many generations of a species, and it's a change of inherited characteristics within a population. And what happens is, over time, those with the most advantageous characteristics will survive to produce offspring, and those are the characteristics that get passed on. And then the last one it gives us is variation. So this is just the genetic differences between individual members of a species. Now we can look back at our sentence. When stem cells specialise to produce gill cells, this process is known as what? Well, looking at our definitions, differentiation is the one that talks about cells specialising into a particular type of cell. So that's the answer that we need to put down. Complete the sentence and choose an answer from the box. So in our box we've got binary fission, mitosis and mutation. And the sentence says, to grow a new gill, stem cells divide by blank. So we've got to figure out which of these words fits. Now, binary fission is the process by which bacteria divides. Mitosis is the way in which most of your body cells will divide. Not to be confused with meiosis, so mitosis produces identical daughter cells, and this is what happens in cells like your skin cells and your muscle cells, and ones like that. Meiosis is something that happens with gametes, and the product cells are not identical. And the last term we're given, mutation, is a change in a gene. So, rereading the sentence, to grow a new gill, the stem cells divide by what? Well, we've got those key words, cells divide, so this tells us it's cellular replication and therefore mitosis. Which one of the following does not contain stem cells? Tick one box. So bone marrow does contain stem cells, and these cells mainly become red blood cells, white blood cells or platelets. They can also become others, so cartilage and bone are other examples, and there are a few more as well. Embryos are formed after the fusion of gametes and some cell replication, and these will be stem cells because they can potentially become a person, and so they begin unspecialised and then they will differentiate into all the cells in the body. Hair is made up of dead cells, so these aren't going to differentiate into anything else later on. meaning they're very unlikely to be stem cells. And lastly, there is meristem. So this is actually found in plants, quite often in the root tips. And these will go on to form more specialised cells in the plants and plant organs. Now we can see that the only one not containing stem cells is the hair, and so that's going to be your answer. Axolotls are small animals. Axolotls are used in stem cell research. What are 2 advantages of using axolotls in stem cell research? Tick 2 boxes. Axolotls are cheap to feed largely because they are so small, and that is good for the lab because it means they have more money for other research areas like equipment. The fact that they are easy to breed is also good for the lab because they can have many of them. But then it says that axolotls are endangered. Well, that's not a good thing about using them in stem cell research. And then they do live in water, but that's not necessarily an advantage. And then axolotl research is cruel. Well, that's also not an advantage, and is more a factor concerning ethics. So the 2 options we have that are correct are the first 2, that axolotls are cheap to feed and that they're easy to breed. Oxygen uptake in humans takes place in the lungs. Figure 4 shows the human breathing system. So here's figure 4, and then we have a diagram labelled A through to E. Where does oxygen enter the bloodstream? Tick 1 box. So if we have a look at the route that the oxygen would take into the lungs, it goes down the trachea, which is E, and then it goes through where A is pointing to, so that's where it splits into the bronchi. Then you go down into the bronchioles and then into D, which points out an alveolus. And the alveolus is in very close proximity to the capillaries and has close access to the bloodstream, so that will be the answer to this question. Name part E on figure 4. So the answer to this is the trachea. Which blood vessel carries blood to the lungs? Tick 1 box. The first option, the aorta, carries blood out of the heart into the body. The pulmonary artery carries blood from the heart to the lungs. And the vena cava carries blood from the body into the heart. So the correct answer here is the pulmonary artery. This question is about leaves. Complete the sentences choosing answers from the box. So in this box we've got epidermis, waxy cuticle, phloem, xylem and palisade mesophyll. So before we have a look at the sentences, let's just check what each of these words actually means. The epidermis refers to the epidermal tissue, which is on the top and the bottom of the leaf, and it can be described as the cells lining the upper and the lower surface of the leaf. The waxy cuticle is a waterproof layer on the very top of the leaf, and it helps to prevent water loss while also being transparent so that light can reach the photosynthesizing pigments inside the leaf. The phloem is the tissue that transports nutrients for the plant, and this is often found in the stem. And then the xylem is often found next to the phloem, and it transports water and dissolved ions. Finally, the palisade mesophyll, so this is the layer of cells that's below the upper epidermis, and this is where you find a lot of your chlorophyll so that photosynthesis can take place. If we have a look at our first sentence, it says the layer of cells lining the upper surface and the lower surface of a leaf is the blank. Well, this lines up really well with our first definition of the epidermis, so that's what you want to put here. The next one wants you to say the part of the leaf where the most photosynthesis occurs. So the answer to this one is the palisade mesophyll, and this is because it's got so much chlorophyll, you know that that's where the photosynthesis is going to be taking place. And the last one is asking you where water is being transported, and we've said that also in our definition, so this one is the xylem. Water is lost through small openings on the lower surface of plant leaves. These small openings are called stomata. Figure 5 shows 2 stomata on the lower surface of a leaf. So here's figure 5 just below, and we can see the holes in the underside of the leaf with structures on either side labelled X. The cells labelled X control the width of the stomata. What are the cells labelled X? Tick one box. So the question's referring to these highlighted bits just here, and these are called guard cells, so they contract or relax to make the width of the stomata, which is the gap between them, larger or smaller. If we just confirm why the others aren't correct, the mesophyll cells are more in the middle of the leaf or beneath the surface, so are nowhere near the stomata at the bottom. Root hair cells are in the roots, and the stem cells in plants are called meristem, and these are also mostly found in the root tips, not right next to the stomata. What is the function of the stomata? Tick one box. So to recap, the stomata is found on the underside of the leaf, and it's that yellow highlighted bit just situated between two guard cells, which contract and relax to make the width of it larger or smaller. So stomata are just little holes, and what they do is allow carbon dioxide into the leaf. How is water lost from the leaf? Tick one box. So here the mark scheme just wants you to tick the right keyword, and that keyword is evaporation. Respiration is one of your energy producing reactions, so glucose plus oxygen gives you carbon dioxide and water. And translocation is the movement of glucose molecules that are made during photosynthesis around to different parts of the plant. A student investigated the volume of water lost from two plants. The plants were different species. Figure 6 shows the student's results. So here's figure 6, it's a graph, and we have two lines plotted on it. So the above one is plant A and the lower one is plant B. The y-axis represents the volume of water lost in centimetres cubed, and along the x-axis we have time in hours, so it spans from 0 to 3 hours. Calculate the difference in the volume of water lost by plant A compared to plant B in the first hour. So to solve this, we have to find the difference in the y values when the x values are 1. So we have a copy of the graph on the left, and the dotted line shows you where time equals 1 is. So we need to mark where that crosses with line A and then with line B. So if we follow along to the intercept at line A, we get a value of y equals 5. Then we do the same for plant B and we get a value of y equals 1. We calculate the difference, so 5 centimetres cubed minus 1 centimetre cubed, and that gives you 4. So that's your answer. What could cause plant A to lose more water at a faster rate than plant B? Tick one box. So you've got to think about your factors affecting evaporation. So these can include temperature, wind speed, and surface area. So the first one says plant A has fewer stomata per leaf. Well, these are less holes for the water vapour to escape out of, so that's not going to be correct. The next one says plant A is smaller. Well, that's got less surface area, so that's not going to make it lose water any faster. Then it says plant A has more leaves, so this is more surface area and more stomata, so actually, yeah, that is going to make it lose water at a faster rate. And then the last one must be incorrect, and it says plant A has smaller leaves, which is similar to it having less leaves in that there's still less surface area and less stomata. After the first 2 hours, both plants were moved to a new room. Suggest one reason why both plants lost water at a faster rate in the new room. So there's a few potential answers for this. The first one that you might go for is that the new room is warmer. This is going to cause a faster rate of evaporation. You could also say that the new room is windier, so the breeze carries away these water molecules at a faster rate. The new room could be drier because the water molecules want to move from that more dilute area into the more concentrated area. They want to spread out. And then the last one you could say is that the new room is brighter, so this will also cause a faster rate of evaporation. Some plants have adaptations to stop them from being eaten by animals. Figure 7 shows part of a holly plant. So this term adaptations is referring to the characteristics of an organism that give it a survival advantage. So in this instance it's referring to the plant's defences against being eaten by animals. Describe one way the holly plant is adapted to stop it being eaten by animals. So we can use this image to help us out. One thing that you might notice is that it's got these spiky tips on the ends, so that's going to make it really uncomfortable for animals to eat it. The other thing that you can see is these berries, so these would be poisonous to eat and they'd also potentially be brightly coloured, and that would warn the animals away from eating it as well because it will indicate poisonous. The mark scheme also allows you to talk about the texture of the leaves, so these are going to be quite tough, and that also makes it more difficult for the animal to physically eat it. Any of these you can give as your final answer, and you only need one of them to get that mark. A student investigated respiration in yeast. This is the method used. Add 5 centimeters cubed of a yeast and water mixture to each measuring cylinder. Add different masses of sugar to each measuring cylinder. Mix the contents of each measuring cylinder gently for 5 seconds. Put the measuring cylinders in a water bath at 25 degrees C, and over the next 20 minutes, record the maximum volume the foam reaches in each measuring cylinder. Figure 8 shows the student's results. So here we have figure 8, and we can see these 4 measuring cylinders. The first one's got 0 grams of sugar, the next one has 1 gram, then 2 grams, and then 3 grams of sugar. And we can see that there's a darker section on the bottom of each and a lighter section on top of the last 3. So the lighter section is the volume of foam, and the darker section is the volume of mixture, which is the same for all of them. Which 2 variables did the student control in the method? Tick 2 boxes. So if we look at this term control, it's talking about the controlled variable. This is the thing in the experiment that you must keep the same. So that's what it's talking about here. And if we have a look at the method, we can see that the first step says add 5 centimeters cubed of a yeast and water mixture to each measuring cylinder. So this is something that's common for all of them. So that's one thing that's kept the same. And then further down, we can have a look at step 4, which is the water bath at 25 degrees C. So that's also something that's common for all of them. So those are your 2 controlled variables. It's the volume of the yeast and the water and the temperature. Table 3 shows the results. So here we've got table 3, the mass of sugar is in the left-hand column, 0 through to 3, and on the right we've got the volume. And remember, this is the volume of this mixture at the bottom and the volume of the foam produced. And it says, what is value x in table 3? Use figure 8. So x is the corresponding value for the cylinder with 2 grams of sugar in it. So here we're back at figure 8, and we can see the one with 2 grams of sugar in it is the third cylinder along. So we just need to calculate the volume of this. And we can see the small divisions, so there's 10 divisions for 10 centimeters cubed. So the volume is up there, and we can see that the closest larger division is at 25 centimeters cubed, and we are 3 small divisions above that, so we are looking for 28 centimeters cubed. In the investigation, the yeast respires and releases a gas which causes the foam to rise. Which gas causes the foam to rise? Tick 1 box. So the answer to this question is your first option, the carbon dioxide. We figure this out by looking at our respiration equation. The products from your oxygen and your glucose are water and carbon dioxide. And of those, carbon dioxide is the one that's likely to be a gas. What conclusion can you make about the relationship between the mass of sugar used and the volume of gas produced? So for this one, we've got a copy of figure 8 on the left to help us out. And if we have a look at it, we can see that the masses of sugar are written along the bottom, and they go from 0 to 1 to 2 and then to 3. So the mass of sugar increases as you go along those measuring cylinders. Then if we have a look at the cylinders themselves and look at the contents, so these volumes go from 5 to 23 to 28 and then to 31. So we can actually see that the volumes are also increasing as the sugar increase, and that's what you can say for your answer. That the greater the mass of the sugar, so like the 3 grams in our final measuring cylinder with the volume of 31, the greater the volume of foam will be produced. Why was no foam produced in the mixture with 0 grams of sugar? So here we can use that respiration equation again. If we have a look at the reactants, we've got glucose and oxygen. They're what you need to get out of it, the water and the carbon dioxide. And the carbon dioxide is the gas producing the foam. So the first term, glucose, is provided by the sugar that they added to each of the measuring cylinders. So without any sugar, in the one with 0 grams of sugar, you're not getting any glucose for the reaction, so respiration cannot occur, and therefore that carbon dioxide that's producing the foam won't be made. Why was the measuring cylinder with 0 grams of sugar included in the investigation? So this is to do with the fact that you need to have a controlled variable. You need to make sure that the thing you're investigating is the only thing affecting your experiment. So by having a measuring cylinder with no sugar in it at all, allows you to have a comparison with what would happen if you didn't have your sugar. The top of the mixture can be covered with a layer of oil after step 3 in the method. Suggest why the layer of oil stops the yeast respiring aerobically. And remember, aerobically means it is done in the presence of oxygen, so there is oxygen available. And because the oil has stopped this from happening, it must be cutting off the oxygen. So that's the answer that the mark scheme is looking for this question. What other substance is produced during anaerobic respiration in yeast? Tick one box. So anaerobic respiration is the type that's occurring without oxygen. So we've got to take that out of our respiration equation. And we get glucose goes to another substance plus carbon dioxide, and that other substance is ethanol. So that's the box that you need to tick. A man has the following symptoms: yellow discharge from his penis and pain when urinating. The man has a bacterial infection. What is the most likely cause of the man's symptoms? Tick one box. So what we can tell from these symptoms is that the man is quite likely to be suffering from an STD, a sexually transmitted disease, and is sometimes called an SDI, an infection instead of disease. And this can help us to limit the list of options that we've got. So measles and salmonella aren't necessarily STDs, we're more looking at gonorrhea and HIV. Now, HIV stands for Human Immunodeficiency Virus, and that V, the term virus, tells us that it isn't a bacterial infection, so that eliminates that one as well, leaving us with the correct answer, which is gonorrhea. The man took a full course of antibiotics. The man's symptoms did not improve. Why did the antibiotics not cure the symptoms? Tick one box. So of our options, we've got the bacteria are immune to the antibiotics, the bacteria are resistant to the antibiotics, and then we've got the man is immune to the antibiotics, and the man is resistant to the antibiotics. So the first thing that you can say about this question is going to be about the bacteria, it's not about the man. So that eliminates the bottom two options. So now the choice is essentially between two key terms. We've got the term immune and the term resistant. Now the term immunity is more to do with the man himself, so he might be immune to the bacteria, if he doesn't get the illness at all, it doesn't affect him. But because we're talking about the bacteria, we're talking about resistance, so that they are resistant to the antibiotic. So it's your second box that you want to tick. Using a condom can stop the bacteria being passed to another person during sexual intercourse. Suggest a different way the man could avoid passing the bacteria on to someone else. So the potential answers for this question are that the man can abstain from having intercourse, and then the other option is that he can maintain his personal hygiene, so washing hands after urinating and similar. A scientist investigated the effect of 3 different antibiotics on 3 different types of bacteria, A, B and C. This is the method used. Grow bacteria A on an agar plate. Put 3 separate paper discs, each containing one of the antibiotics, 1, 2 and 3, onto the agar plate. Put the agar plate into an incubator for 48 hours. Repeat steps 1 through 3 for bacteria B and for bacteria C, and figure 9 shows the scientist's results. So here we have figure 9 showing the 3 agar plates, and we can see the first one is the agar plate containing bacteria A, then we have bacteria B, and then bacteria C. And we can see that the greater grey circle is where the bacteria is growing, the white circles are the areas where the bacteria are killed, and then the black points are the paper discs, and the numbers indicate which antibiotic is which. Compare the effectiveness of the 3 antibiotics at killing the different types of bacteria, and this question is worth 6 marks. So to be able to get full marks on this question, you've not only got to be able to make the correct points, you've got to link the relevant points in a logical sequence. And wherever there's a red tick, you know is a relevant point. The first thing you want to look at when tackling this is figure 9 itself, and what you can make for your first points is a remark on which antibiotic is the most effective in each agar plate. And you could also be looking for any particularly ineffective ones, so numbers with very small circles around them. So the way that you actually state this is by sequencing them in a logical order. So you say on bacteria A, 3 is the most effective. On bacteria B, 1 is the most effective, and by most effective we mean the one that has the biggest white circle, the biggest area of killed off bacteria around it. And then on bacteria C, number 2 is most effective. And then you can go on to talk about your least effective ones. So number 3 has no effect at all on bacteria C. And note that all of these sentences are about the physical effects, so these are what we call qualitative observations. And to get your full 6 marks, you want to talk about qualitative and quantitative, which we'll get onto next. So now you want to start to compare the effectiveness, because that's what the question's asking you to do. So you might look at the biggest one, which is 2, and comment that 2 kills more on C compared to how much bacteria kills on A or B, but it kills about the same amount for A and for B each, because if you look at the bacteria A, so the first agar plate, and the bacteria B, the second agar plate, you can see that the values for 2 and 2 are quite similar. The circles are about the same size. Then if you go on to talk about number 3, you can follow the same kind of structure with your explanations. So 3 kills more on A than on B or C, that's its biggest white circle on bacteria A, and it kills more of B than it does of C, because if you look at the second one along the agar plate containing bacteria B, you can see that the circle for 3 is there, whereas on bacteria C there's no white circle at all because it doesn't work. So you can say that C is resistant to 3. And if you go to talk about number 1, you can say that 1 kills more on bacteria B, so that's the second agar plate along, than on A or C, and it kills more on C than it does on A. You can see that on the first agar plate with bacteria A, the white circle around number 1 is very, very small, whereas on bacteria plate C, it's got a slightly larger white circle. So these comparative statements, like 2 kills more on C compared to A or B, these are quantitative because you're comparing the more or less values. So to get the full marks, you're talking about the qualitative effects and the most effective or the least effective, but then you're also talking about the quantitative comparisons. Milk contains bacteria. A small volume of raw milk was placed in a counting chamber in a special type of microscope slide. Figure 10 shows what the counting chamber looked like when viewed using a microscope. So here's figure 10, and a single bacterium is represented by one of those small black dots on a 0.1 by 0.1 millimeter square. A scientist counted the number of bacteria in 4 samples of raw milk. Table 4 shows the results. So here we have table 4. The left-hand column is the milk sample from E through to H, and the right hand is the number of bacteria in the counting chamber. Which milk sample is shown in figure 10? Tick one box. So in table 4 we have the number of bacteria in each milk sample. So what we've got to do is count the number of bacteria in figure 10. So we count the small dots, we've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. Looking back at table 4, we can see that the one with 15 is milk sample E, so that's the box that we tick. Calculate the mean number of bacteria in the 4 samples in table 4. So to calculate the mean, we have to get the sum of the values that we're looking at, and then we have to divide by the number of values that there are. So you calculate 15 plus 12 plus 13 plus 16, and you get an answer of 56. Next we see how many values we've got, so 15, 12, 13 and 16, that's 4 values. So we take off 56 and we divide that by the 4, the number of values, and the answer to that is 14. So you'd get 1 mark for your calculations and another for your correct answer. Calculate the mean number of bacteria per millimetre of milk in the samples. Complete the following steps. Calculate the total area of the counting chamber in figure 10. So looking at figure 10, it's a square and it's 0.1 by 0.1 millimetres. To define its area, we've just got to multiply those sides together. So 0.1 multiplied by 0.1 is 0.01 millimetres squared. The depth of the counting chamber is 0.01 millimetres. Calculate the volume of the counting chamber in figure 10. Use the equation volume equals area multiplied by depth. So we've got the area that we just calculated, that's 0.01, and the depth we've been given is also 0.01. So we multiply them, 0.01 by 0.01 gives you an answer of 0.0001. And that's your final answer here in millimetres cubed. Calculate the mean number of bacteria per millimetre cubed of milk in the samples. Use the equation mean number of bacteria per millimetre cubed of milk equals the mean number of bacteria divided by the volume of counting chamber. We calculated the mean number of bacteria previously and we've got an average of 14. And then the volume of the counting chamber was also calculated as 0.001 millimetres cubed. Now we've got to take the 14 and divide that by our 0.001 millimetres cubed, and that gets you an answer of 140,000. Milk is heated to reduce the number of bacteria it contains before it is sold for humans to drink. Milk with more than 20,000 bacteria per centimetre cubed cannot be sold for humans to drink. Table 5 shows the number of bacteria per centimetre cubed in 4 different samples of milk. So here we have table 5, on the left is the milk sample from P to S, and the number of bacteria per centimetre cubed of milk is on the right. Which of the milk samples could not be sold for humans to drink? Tick one box. So the questions told us that more than 20,000 bacteria per centimetre cubed of milk is dangerous for human consumption. However, when we look at the values that we've got in the table, we see that they are in standard form, which isn't the case with our 20,000. So what we've got to do is convert the 20,000 into standard form so that we can compare it. We make a decimal point after the first number because the first number has to be between 1 and 10, and then we count how many spaces there are afterward to get our power of 10. In this instance we've got 4, so we're going to get an answer of 2.0 times 10 to the power of 4. That's our 20,000 in standard form. So if we have a look at the table and see what is larger than this, 1.8 times 10 to the power of 4 is less than 2 times 10 to the power of 4. However, 2.2 times 10 to the power of 4 is greater, so Q would be our answer here. The reasons the last two are incorrect are in the powers of 10. So the next one beneath is 2.2 times 10 to the minus 5, so that tells you it's going to be a decimal, so that's a small number. And then the next one is 1.8 times 10 to the 3, and times 10 to the 3 is a smaller value than times 10 to the 4. So the correct answer is definitely milk sample Q. Why should milk sold for humans to drink not contain large numbers of bacteria? So the answer that the mark scheme is looking for you to say here is the fact that the bacteria can make humans ill. It releases toxins that can make you sick or they can kill people. Figure 11 shows the internal structure of the human heart. So here we have figure 11 and the heart diagram is just below. Which organ system is the heart part of? So the answer to this question is the circulatory system. Draw a ring around one valve on figure 11. So here's figure 11 and this here is a valve. So you can think of them as just gaps between the chambers of the heart. So this diagram actually shows 4 and they all have their own names. The first one we circled is called the tricuspid valve. On the right is the mitral valve, then the aortic valve which leads through to the aorta, and lastly the pulmonary valve. What is the function of the valves in the heart? So the function of a valve is to prevent the backflow of blood. It stops it from going in a direction that it's not supposed to go in. Valves are also found inside some blood vessels. Which type of blood vessel contains valves? So the answer to this question is veins. Sometimes a valve in the heart can begin to leak. A leaking heart valve may be replaced with either a mechanical valve or a biological valve from a pig. Table 6 shows information about the replacement valves. So here's table 6. On the left is the properties of the mechanical valve. So it's made of plastic or metal. It can cause the blood to clot around the valve, but there's no need for a replacement valve within 5 years. On the biological side, on the right, it's made from living tissue. There's no risk of blood clotting around a valve, but sometimes another replacement valve is needed after 5 years. Suggest 2 reasons why a patient may choose a mechanical valve and not a biological valve from a pig. So the advantages of a mechanical valve, as shown in the table, was that it was less likely to wear out after 5 years, so it's longer lasting. The other advantage is because it's mechanical, the body's not going to have an immune response to foreign biological tissue. This means that the body is not going to try and reject it. The patient doesn't have to take any anti-rejection medication or any immunosuppressants, and these drugs they would have had to take for the rest of their lives. So you only need 2 points to get those 2 marks, however there are some alternatives. So you can just point out that the risk of rejection is lower. You can also talk about the ethical concepts. So some people may have religious or ethical concerns about taking the valve from a pig which can't give its consent. So any of these points is relevant and will get you one of those 2 marks. Suggest one reason why a patient may choose a biological valve from a pig and not a mechanical valve. So the answer to this one you can get from the table, and that is that the biological valve does not have a risk of clotting in the way that the mechanical valve does. So the patient will not have to keep taking anti-clotting medication, and that's the answer that you need to put. A person may develop other medical conditions. Draw one line from each medical condition to the correct treatment. So the 2 medical conditions that we've got on the left are high blood cholesterol and irregular heartbeat. So high blood cholesterol is dealt with by statins, and what these do is lower the cholesterol levels inside the blood. Irregular heart rate is treated with an artificial pacemaker, and the key for this one is that it's called a pacemaker, so it corrects the pace of the heart. In the case of the other 2, the antibiotics would be used to treat a bacterial infection, and insulin is used to treat somebody with diabetes. Figure 1 shows an animal cell viewed using a microscope. So here's figure 1, and just below you can see the cell. The question says the cell contains a nucleus. What is the function of the nucleus? So looking back at figure 1, the nucleus is this part down here. It contains the DNA of the cell and all the instructions it needs to be able to carry out reproduction and growth in the future. And so the way you can say this is that it controls the activities of the cell and it holds its genetic material. So either one of those statements, controlling it or holding the genetic material, will get you that mark. Name one type of cell that does not contain a nucleus. So there are a few answers you can potentially give here. One is the red blood cell, and this doesn't contain a nucleus because it needs that room for hemoglobin. Alternatively, you could mention bacteria or any other kind of prokaryote, and these don't have a membrane-bound nucleus. Instead, they have their chromosomal DNA free in the cytoplasm, and they can also have loops called plasmids, which contain genetic material. And then you can also mention the xylem cells, so those would be in the plants. And they don't have a nucleus because they're not living cells, they're dead cells just acting as a tube to carry water, and so they're not carrying out any functions that a nucleus might be needed for. Draw a simple diagram of the cell in figure 1. Label 2 parts of the cell. So if we have a look back at our figure 1, we've already got some key components. We've got that general outline over there, which is going to be the cell wall, and we've also got the nucleus, which we mentioned earlier. So what the mark scheme is looking for you to do is draw the approximate same shape as the cell we see before. You can label the outline as the cell membrane, so that controls what goes in and out of the cell. We can also draw the nucleus, so that was quite a clear component in the diagram, and that controls the activities of the cell. So both of those will get you a mark and you only need 2 components. Alternatively, you can label the cytoplasm, so that's where your chemical reactions might be occurring in the background. You can label a mitochondrion, the site of respiration in the cell, and you can also draw a ribosome, so this would be smaller than the mitochondrion, and it's the site of protein synthesis. Name one structure found in a plant cell but not found in an animal cell. So there are a few potential answers here. You could mention the cellulose cell wall, which isn't needed so much by the animal cells because animals have skeletons, whereas the plants need that cell wall for support and keeping it rigid. Alternatively, you could mention chloroplasts, so these aren't needed by animals because animals don't photosynthesize, plants do. And then you might also mention the vacuole or the permanent vacuole, and these are needed more by plants than by animals because they contain water and cell sap and they help the plant maintain rigidity, whereas an animal cell doesn't need to be so rigid or under such pressure. Figure 2 shows some different cells. So here's figure 2 below, and we've got a group of cells and we have this label X through Y. The question says the real length from point X to point Y is 0.06 millimeters. Calculate the magnification. And it gives us this formula we need down here. So magnification equals size of image over real size of object. And the question is worth 3 marks. So what you want to do here is get your ruler and measure this length from X through to Y, and then just record that value, because this is how you're going to get your image size. And it would have been printed to a specific size on the paper. So in this instance, the size you would have measured is 24 millimeters. And the reason we do it in millimeters as opposed to recording it in centimeters is because the question gives it to us in millimeters, and so this saves us having to do that conversion. So back to the question, we've now recorded our size of image, so that was our 24 millimeters, and then the real size is given to us by the question, that's the 0.06 millimeters, and then we just plug that into our equation. So 24 over 0.06 will give you the magnification. That will tell you how many times bigger the microscope has made the image. So 24 over 0.06 gives you an answer of 400 times. And your marks would be for making that measurement of the 24 millimeters, putting your equation together correctly, and then getting that final answer. The cells shown in figure 2 were viewed using a light microscope. Give 2 advantages of using an electron microscope instead of a light microscope. So electron microscopes work by firing a beam of accelerated electrons at the specimen, and then these get reflected back and detected by the microscope to create an image. The electrons actually have a shorter wavelength than light does, and this means that they can get into those spaces and detect the smaller features, which means that the electron microscope therefore has a higher resolving power, so it's better at telling the difference between 2 small objects and identifying that they are 2 things rather than just one, and it also has a higher magnification power. So those last 2 are what you can put for your answer. You can say that it has a higher magnification, and then you could say that it has a higher resolution or a higher resolving power. Another thing you can potentially say is that it gives you a 3D image, and this is because with the electron microscope, as opposed to just looking through the specimen as you are with a light microscope, this one's actually detecting the electrons that are reflected off the surface of the specimen, and that gives it a perception of depth. Mosquitoes carry a pathogen that causes malaria. What type of pathogen causes malaria? Tick 1 box. So the answer to this one is a protist, and specifically it is called the Plasmodium protist. Mosquito nets can help prevent the spread of malaria. Table 1 shows the results of a study in one area of Africa. So here's table 1, the left-hand column says the total number of people in the study, so that's everyone involved. The next one is the number of people who use mosquito nets when sleeping. And then the next one along is the percentage of people with malaria, and it's divided into those who use mosquito nets when sleeping and those who do not use mosquito nets when sleeping. A newspaper made the following statement: "Study shows mosquito nets are scientifically proven to prevent malaria. Give 1 piece of evidence that supports the statement." So we want to be looking at this section of the table over here, the percentage of people who do have malaria. And we notice that the percentage of people with malaria who do use mosquito nets is only 1.2. Those who do not is 40%. So this shows us 1.2 is a much smaller percentage than 40%. So those who use nets make up a smaller percentage of those with malaria. And that's the kind of statement that the mark scheme is looking for you to make, that there is a lower percentage of people with malaria when they're using nets. Suggest 1 reason why the statement may not be valid. So there are a few answers you can potentially give here. One is that when you look at that table, some people who use mosquito nets do still have malaria. It's not 100% effective. Another answer you could give is that the data is only for one area of Africa, and you don't necessarily know if the trend is the same everywhere. Another thing you could say is that the group size is too small and you perhaps don't have enough data to draw that conclusion. And lastly, you can argue that no other information about the people in the investigation is considered, so there could be other factors regarding whether you get malaria or not. Table 2 shows information about the number of deaths from malaria in the same area of Africa. So here we have Table 2. The left-hand column is the year and it goes from 2005 through to 2015. Then the right-hand column is the number of deaths from malaria per 100,000 people. Predict the number of people per 100,000 who died from malaria in 2017 if the trend stayed the same. So here we want to start by having a look at the trend over the last few years to see if we can predict what's going to happen in the next 2 years to get to 2017. So from 2011 to 2013, the number of deaths per 100,000 decreases by 3. Similarly, from 2013 through to 2015, it goes down by 2. So the next jump we want to make wants to be decreasing by 2 or 3. So as an example, we might decide to make the prediction that the number of deaths from malaria per 100,000 people will decrease by another 2 in the next 2 years. So we will take our current value. In 2015, 92 people died from malaria per 100,000, so that's our starting value. We take away 2, as is our prediction for the next couple of years, and that gives us a value of 90. So we might say that in 2017, 90 people per 100,000 will die from malaria. Now remember, this is a prediction that we're making. The trend we've seen so far has been a decrease of 3 and then a decrease of 2. So you as the candidate might decide to predict that it's going to stay around -3 or -2, or you might follow that decrease and go for -1. And because of this, the mark scheme has been written to allow any value between 88 and 91, because we are making a prediction and there isn't a certain number. But the value we calculated in our example, the value of 90, is perfectly within those parameters, so you'd get a mark for that. Use of mosquito nets has helped to reduce the number of deaths from malaria each year. Suggest one other reason for the reduced number of deaths from malaria each year. So there are a couple of answers you can put here. One is improved healthcare. Another is the use of mosquito control methods. And then there can also be different behaviours among people, so changing the way that people act so that they get bitten less often. Describe how the human body prevents pathogens from entering and defends itself against pathogens once those are inside the body, and it's worth 6 marks. So when going for full marks on a question like this, you've got to make sure that you're making your points in a logical order, and then you've got to explain them so that the examiner really knows and you understand what you're talking about. And then you've also got to make sure you include both of the bullet points given to you in the question. So let's start by considering the first bullet point, how the body prevents pathogens from entering into it. The first thing that you could talk about is the skin. So this acts as a physical barrier, and your keyword here is barrier, so make sure you mention that specifically. Next, you might go on to talk about this oil on the surface of the skin, and this has the property of repelling bacteria and pathogens. It's also worth noting that this oil can be called in the mark scheme sebum, so if you see that, it just refers to the oil on the surface of the skin. Now you can start to talk about some other forms of protection. So when the body gets a cut or a graze, a scab will form over the top of it, and this also forms a barrier to stop the pathogens coming in direct contact with the bloodstream. You can also mention that the eyes produce tears, and these tears are antiseptic, so they will kill any pathogens trying to enter. And you can get a mark for either of those, saying that the eyes produce tears and then mentioning that they're antiseptic. And then the eye also contains an enzyme which will kill bacteria, so that's another feature that you could mention. Then there are marks available for you talking about your breathing system, so all to do with your nose and your trachea and your bronchi. And what you can say about these is that they contain mucus. Mucus is a sticky substance which will trap the pathogen and stop it from going all the way into your body. The mucus is then carried away by the cilia, so that can be carried back up so that you can spit it out, or it will go down into the stomach to be dealt with by the hydrochloric acid. And the term cilia is just referring to all these small hairs that line your air passages, so your nose and your trachea and so on. So overall you can see there are multiple marks available for the prevention of entering, but just remember that just listing 6 marks or 6 bullet points isn't necessarily going to get you all 6 marks, so ensure they are logically ordered and explained well, and you're including both of the bullet points. So we can move on to the second one now, defending against pathogens once they're inside the body. So when a person is infected with a pathogen, one of the first things that's going to happen is the immune response, and this is caused by your white blood cells and your immune system, and so that's the first mark that you can go for the second part of the question. Then you might go on to talk about the production of antitoxins, so this is done by the white blood cells, and the antitoxins will neutralise the toxins that are released by the pathogens, so it's battling the parts that actually make you feel ill. Similarly, you can talk about the production of antibodies, and these are responsible for destroying the pathogen itself, so those statements will get you another couple of marks. Then lastly, you can talk about what's called memory cells, and these are a part of your immune response, so they recognise a pathogen that you see beforehand, and then should that pathogen re-enter the body, it will trigger a more rapid response to it, and it will help you to recover from the infection faster than you would have initially. This question is about photosynthesis. Complete the word equation for photosynthesis. So on the side of the reactants you've got carbon dioxide and water, and then on your products you've got glucose. A student investigated photosynthesis using pondweed. Figure 3 shows the apparatus that the student used. So here you've got figure 3, you've got some pondweed and it's situated in a beaker of water under a funnel, and the funnel is guiding up bubbles into a measuring cylinder. So those bits that are highlighted in yellow are the bubbles, and the measuring cylinder catches these and effectively measures the gas that is being produced by the pondweed. This is the method used. Set up the apparatus as shown in figure 3. Switch on the lamp. After 20 minutes, record the volume of oxygen collected in the measuring cylinder. Then repeat steps 1 through 3 using bulbs of a different power output. What was the independent variable in the investigation? So first let's just be reminded of this term independent variable. So this is the variable in the investigation that you are changing. And the methods told us what this is because the last step said repeat the steps using bulbs of different power outputs, so that's what's going to be different each time, so we tick that top box. Suggest 2 ways the method could be improved so that results would be more valid. So there are several options that you could go for this one. One might be doing repeats and then calculating a mean value. Another might be controlling the temperature of the water that your pondweed is in. And you only need 2 points to get those 2 marks, but there are some alternatives as well. So you can make sure that you're controlling the distance between your pondweed and your lamp, and this can have an impact because of inverse square law. Then you can be controlling all of your factors to do with the pondweed itself. So for example, how old it is, how long it is, what the species is, what the mass is, just making sure it's the same that you're using for each part of the experiment. And then lastly, you can be giving it time to equilibrate, which means just getting it used to that new light level that it's at. Table 3 shows the students' results. So here you've got table 3, the left-hand column is the power output of the bulb in watts, and then you've got your central column of the volume of oxygen collected in 20 minutes. Then the last column is the rate of photosynthesis in centimetres cubed per hour. And the question wants you to calculate the value of x in table 3. So here is x in the n column. We're calculating the rate of photosynthesis in centimetres cubed per hour for a bulb that has a power output of 150 watts. So looking at this, we've got our known value of 1.1, and that's the volume of oxygen collected in 20 minutes. So that's 20 minutes worth of photosynthesis, and we're looking for x, which is an hour's worth of photosynthesis. So how many sets of 20 in that 60? Well, that's 60 over 20, which is 3. So 1.1 multiplied by 3 is 3.3. That's how many centimetres cubed are produced in an hour. So that's the answer you're going to get there. Complete figure 4. You should label the x-axis, use a suitable scale, plot the data from table 3 and your answer to the previous question, and then draw a line of best fit. And the question is worth 4 marks in total. So here we've got figure 4, it's an empty graph and we have to complete it using the table on the left hand side. And in order to do that, you've got to fill in your x-axis. So as a rule, the independent variable will always go along the x-axis, and the dependent variable, so that's what you're measuring, will go onto the y-axis. In this instance, the independent variable is the power output of the bulb in watts. That's what we're changing as we go through our experiment. So we want to fill that in at even intervals along the bottom of the graph. So our smallest value is 60, and then we've got 100, 150, 200 and 250. So most of them go up in 50s, and if we test that out on the scale of our graph, we can see that that actually works out quite nicely for us. Just don't forget to specifically add that label of power output in watts. Okay, so now you can actually start to plot your points. So the first one will have an x value of 60, and then it will have a y value of 1.5 because it's the rate of photosynthesis in centimetres per hour that we're going up in. Make sure not to accidentally start plotting the values for volume in 20 minutes. So we work our way up from the value of 60 on the x-axis, and then on the y-axis we plot along at our value of 1.5, and then we make an x where they cross. So you just repeat that process for each of the points. So the next one will be 100 and 2.4. Then your 150 and 3.3, 200 and 3.6, and then 250 and 3.6. Once you've got all of them down, you then do a nice smooth curve of best fit, and it should go through all of your points. Determine the expected rate of photosynthesis with a bulb of power output 75 watts. Use figure 4. So what you've got to do here is find the value of 75 watts along our own scale. So 75 watts will be right between the 50 and the 100 on our own scale. And once we've got that, we work our way upwards until we intercept our line of best fit, and then where that crosses, we then find the corresponding y value, and that will give us our answer. So in this instance, we're getting 1.9, or just a tiny bit below it. So we can put that down for our answer. And for this question, the mark scheme will allow you a little bit of leeway, plus minus a small square. So if you got 1.8, that would also be accepted. Which graph shows the effect of temperature on the rate of photosynthesis? Tick one box. So to be able to answer this question, there's one bit of key knowledge that you need to have. And this is that as temperature increases, the rate of photosynthesis will also increase. However, this only happens up until a certain extent, because eventually the temperature becomes high enough that it's actually doing damage to the plant. Things like dehydration and denaturation can occur. So when this is plotted on a graph, you will see the rate of photosynthesis rising upwards and then suddenly decrease at a point. And that is the third graph down, so you tick that box.